Hi there. This is going to be a short video on the Module 5 topic, DNA and protein synthesis. To get full benefit from this video, you'll need access to a set of my notes and a set of questions, and you'll have a set of answers as well. I'm going to go through the topic as quickly as I can. And as I go through the topic and different areas of it, I'm going to refer to questions, and I'm going to, as far as possible, attempt to go through the relevant question dealing with each part of this topic. So the main parts of this topic are, first of all, the nature of the DNA code itself, and then the other parts would be transcription, how the DNA code is copied onto messenger RNA, and then translation, how the messenger RNA is used in the process of making a protein, which is translation. And then finally I'm going to finish on the area of epigenetics, which is how the DNA code can be, as it were, modified in translation. Okay, so I'm going to move through this topic as quickly as I can. I'm going to some of the easier areas, some of the lesser important areas, I'm going to go through very quickly. In other areas, I'm going to slow down and go through a bit more slowly. Let's get started. At GCSE, you'd been asked the question, what is DNA code for? And the answer was the DNA codes for proteins. DNA is a code for proteins. So the DNA that's present inside a cell and present inside the nucleus is a set of instructions, as it were, telling the cell how to make its proteins. Cells make lots of proteins. In fact, it's a major activity inside a cell, making proteins. Major proteins a cell would make would be enzymes. Enzymes are a very important group of proteins that cells are making. Other proteins that cells have been making might be cell membrane proteins, like channel proteins and aquaporins and active transport proteins and so on. So those are important proteins as well. And then you have other specialized individual cells, individual specialized cells that are making very specific proteins. So for example, B lymphocytes, plasma cells will be making antibodies. They're antibodies are proteins. Cells maybe also on the surface of the cell you have antigens, which are proteins as well. And then other cells might be making hormones. Many hormones are proteins. So for example, the hormone insulin is a protein, and it's made by pancreas cells. And then you have other proteins that are made specifically in certain cells, like hemoglobin is a very important protein. Anyway, this topic is basically looking at how the DNA and the proteins are interrelated. And so we're going to start off, first of all, just saying a wee bit about the DNA code. The DNA is basically a double-stranded molecule made up of two side strands, and then with base pairs down the middle. And it's the base pairs that are important in determining, basically the base pairs are the code. The base pairs determine the protein that's produced. Then you might ask the question, how exactly do a sequence of base pairs or a sequence of bases in a protein determine or how are they related to the production of a protein? Well, remember that a protein starts off, the production of protein involves firstly the production of a polypeptide, and a polypeptide is a sequence of amino acids, and that sequence of amino acids can be very variable because you have 20 different amino acids to deal with. And so when you're making a polypeptide, you're joining up amino acids in a very precise sequence. And the precise sequence of any polypeptide is going to determine the finished protein. So when a cell's making a protein, the cell needs some instructions, as it were, or some detail, some plan as to the order in which it should join its amino acids together. And that's what the DNA does. The DNA is a code or a set of instructions coding for the sequence of amino acids in a polypeptide. And you could say it another way, you could say the DNA is a code for the primary structure of a polypeptide. And that primary structure then determines everything else about the protein. The primary structure is right, then the protein is right, the structure of the protein is right. And protein structure is very important. Most proteins are dependent on their shape, the shape of the protein. So for example, enzymes are very dependent on their shape, antibodies are dependent on the shape, channel proteins depend on their shape, active transport proteins, shape is very important for proteins. So the order of the amino acids in the polypeptide at the start determines the shape of the protein. If you get a mistake in the polypeptide, get a mistake in the sequence of amino acids, then you have a mistake in the protein itself. Then won't be correct. It won't be the correct shape. It may be non-functional, may not work at all. So let's look at this the DNA code then. You'll see on page 2 there I've illustrated that the code works on the basis that each 3 bases. Well, first of all, you can recognize that the 2 strands of the DNA, one of the strands is called the coding or template strand, or even the sense strand, and the other strand of the DNA is called the non-sense strand or non-coding strand. So in making a protein, you only need one of the 2 strands of the DNA, and that's called the coding strand. And in my picture there, it's the strand on the right. And you'll see that each 3 bases code for a different amino acid. So the first 3 bases there, AGG, code for the amino acid serine. And that sequence AGG is referred to as a DNA triplet, a triplet of bases. So that triplet of bases codes for serine. And then you see the next triplet of bases, GCA, is a triplet for the amino acid arginine. And of course, when you get your amino acids then lined up in order, you can join them together by peptide bonds to make a polypeptide. So the first aspect of the DNA molecule is that it's a triplet code, or its code is a triplet code. In other words, it takes 3 bases to code for each amino acid. If you just use 2 bases to code for each amino acid, then you'll see there at the base of page 2 that I've listed all the possible codes you could have. Each it would be called a doublet code. Each 2 bases would code for an amino acid. Well, the problem with that is that you only have 16 different possible combinations of each 2 bases, considering you have 4 bases, A, C, T, and G. Any 2 of those gives you an amino acid. That wouldn't work because you'd only have 16 codes. And 16 is not good enough because we need 20 codes, because there are 20 amino acids. So when you go to having 3 bases coding for each amino acid, you'll see that there are 64 possible different sequences of 3 bases taken from 4. And 64 then is lots of codes, lots of different triplets. And for that reason, what you find is that the DNA code, you find that several different triplets code for the same amino acid. So each amino acid may have several triplets. The example I've given there is valine on page 3. I've said valine or valine, the amino acid valine can be CAA, CAT, CAC, or CAG. Any one of those 4 occur in the sequence, you get a valine molecule, a valine amino acid in the sequence of the polypeptide. I've said there for most amino acids, it's only the first 2 bases that are important, and you'll see in that case, the first 2 bases are always CA, and once you have CA, you always get valine. The 3rd base can be any base. I've said suggest an advantage of this. Well, the advantage of this is the 3rd base can be any base. And sometimes you get mistakes in the code, and basically what I'm saying here is that if you get a mistake in the 3rd base, it doesn't matter. So instead of, say, the 3rd base, if the 3rd base should be normally adenine or A, and by mistake it becomes thymine, then that doesn't matter. You still get the same amino acid. By the way, mistakes in the base sequence are referred to as mutations. We'll look at mutations later on. The next important bit is the DNA code is non-overlapping. That means when you're reading the code, you read the first 3 bases and that codes for the first amino acid. Then you move on to the next 3 bases and that codes for the next amino acid. If the code was overlapping, in other words, when you use the first 3 bases for the first amino acid, you could use one of the first 3 bases in the second amino acid. Well, if that was the case, then that might possibly mean that if there was a mistake in a base, that mistake then would affect 2 amino acids rather than just the first amino acid. So one of the advantages of the code being non-overlapping is that it gives the code a certain amount of mutation proofing. Okay, moving on then quickly then, the DNA code is a universal code. That simply means that the DNA code, wherever you discover it, wherever you find it in any living organism on this planet, the same sequence of bases always code for the same amino acids. So it's the same code everywhere. Item number 5 there is very important. Much of the DNA code is non-coding DNA. now we're taught at GCSE the DNA code is very important, and we're even suggesting now the DNA code is very important. And then to be told that most of the DNA code is actually unimportant, as it were. It doesn't code for any amino acids, and that only approximately 2% of the DNA in on the DNA code is significant and actually codes for proteins. The non-coding DNA is referred to as introns. So regions of non-coding DNA are referred to introns. Regions of coding DNA are referred to as exons. Now we want to look at this idea of a gene. A gene then is just a length of DNA, a short length of DNA. And the idea would be, the simplistic idea is that a chromosome sort of looks like that. And then the DNA, this is a very simplistic picture of DNA and chromosome. The DNA then just is a molecule that's wrapped around the chromosome. So this is the DNA molecule. So there's the DNA molecule wrapped around the chromosome. Now, what then is a gene? Well, there might be a gene, that little short bit at the end, and then there's the next gene, and then there's the next gene. So genes are just little sections of DNA. So as you move up the DNA here, you just have like maybe 1,000 genes on that one chromosome. So genes are just short sections of DNA of a certain length. And each gene, as we'll discover later, codes for a polypeptide. There's a theory that says that one gene codes for one polypeptide. So I've just said there that we just want to explain this idea about the exons, the coding and the non-coding regions. And you'll see at the bottom of page 5 there, I've drawn a chromosome, and I've shown some DNA coming off the chromosome, and I've said you've got different bits of DNA or different genes. So you have one gene, then you have the next gene. You'll notice the gene. Notice the gene at the very bottom there. And I particularly did this picture just so they could do that particular gene there. And basically the idea of drawing that gene is just to show you that within that gene, if you look closely, you can see that there are, and at the start of the gene here, you've got an exon, which is a coding piece of DNA, and then you have an intron, which is non-coding, then you have an exon, then you have an intron, and so on. And so what I'm trying to get across to you there is that while genes are the important components, they code for the proteins. Even within a gene, even within the sequence of the gene, you find non-coding bits, introns. So you have coding bits and non-coding bits, even within a gene. So your thought process might be that maybe all the coding DNA is inside the genes, and the non-coding DNA is somewhere else. But actually, no, it's not. It's the non-coding DNA is sort of interspersed among the coding DNA, mixed in among the genes. We're not certain what the role of the non-coding DNA is. It's thought to have some sort of regulation, some way or another, maybe regulating which genes are switched on and which genes are switched off, and so on. Now, questions we want, maybe just look at a couple of questions here just for a second. First of all, question 1C is asking us about, it says question 1C, the code is degenerate. So we'll answer that question. DNA code is degenerate. Explain what's meant by this, the term degenerate, and identify where in the diagram a degenerate code is illustrated. Well, we'll just do the first part of it. So the idea of a degenerate code simply means that each amino acid is coded for by more than one DNA triplet. And that's the definition of a degenerate code. We'll come back to the rest of that question later on. I just want to look then at a wee bit about the coding and non-coding DNA. So if you look at question 3C, there at the introns and exons, you'll see in question 3C, DNA lengths measured in base pairs. Okay. It then goes down, it says, how many base pairs were required to code for an amino acid? Code for 84 amino acids. So if you have a polypeptide that's 84 amino acids long, that's a lot of amino acids joined together to make the polypeptide. Then it's kind of asking how many bases, how many base pairs are required for that. Well, a base pair would be an A with a T and a C with a G and so on. And the answer would simply, you would multiply 84 by 3, because it takes 3 bases to code for each amino acid. So that would give 152, I think. 384 is 152. So the answer to part 1 is 152. And then it tells you that the actual gene coding for this polypeptide is 330 base pairs long. So you only need 152, but the actual gene is 330 base pairs long. So it seems to be a lot of extra bases there. And the answer is that the extra base pairs are all non-coding DNA. That's introns. So in among the, it only takes 152 bases long to make to code for the polypeptide, but there's a lot of extra bases there, and that's non-coding DNA, and that's the answer to that question. Question 3C part 2. You'd say it's the rest of the bases are non-coding DNA. Okay, so we want to move on then quickly to this idea, the one gene one polypeptide theory, and I'm going to refer you to question 3B here. The one gene one polypeptide theory, as it says a gene is a length of DNA with sufficient codons to code for a single polypeptide. Originally, the idea, there was an original theory that said each gene codes for a protein, but they've now realized that can't be the case because proteins may have several polypeptides, and each polypeptide may be different. So you can't have this one single piece of DNA, one single piece of code, one gene coding for all those different polypeptides. It simply doesn't work. So you'll see that's what it says there. It says, use your understanding of protein structure, suggest why the one gene, one polypeptide idea or hypothesis is more accurate than the earlier versions, which were one gene codes for a protein or one gene codes for an enzyme. So the first bit is you're going to say, well, the idea of one gene coding for a protein doesn't really work because proteins may be made up of several different polypeptides, and so the one gene couldn't code for all the different polypeptides. And one gene, one enzyme. Well, the answer to that basically was that a gene doesn't always code for an enzyme. A gene can code for other proteins. So the answer to that second part is say, not all genes code for enzymes. Genes code for all sorts of proteins. And explain precisely what's meant by the term one gene, one polypeptide. And you basically say a gene is a length of DNA with sufficient base sequence to code for the precise structure of the amino acids in a polypeptide. So you've got that definition at top page 6 there. A length of DNA that codes for the sequence of amino acids in a single polypeptide. Now, so that's all of those bits with the DNA code. Now, briefly, I'm going to talk then about mutations. Now, some teachers don't talk about mutations in this section. They leave it to later on in the specification. In fact, it's mentioned later on in the specification, and I will do it there as well. But gene mutations, I'm going to talk about it briefly. I'm going to make reference there to question 1D is really the only question in this booklet here that makes specific reference to mutations. Question 1D. So what are mutations? Mutations are mistakes in the base sequence of a gene, and there's two kinds of mutations. There's one called base substitutions and one called base deletions. Basically, a base substitution is where a base in a sequence is replaced by a different base, replaced by the wrong base. An example I've given there is that in the correct sequence, the base the code, the base sequence is CTC, and so on. You can see that that codes for glutamic acid. But the mutation that's happened in this case is that the T base has been replaced by an A base, and you can see the effect of that. The T base has been substituted by an A base, and the result is that you don't get the amino acid glutamic acid, you get a different amino acid, valine. So you can see how a single base sequence wrong in the sequence in the gene can give you a different amino acid. So in this case, you get one wrong amino acid in the sequence, and you might say, well, in a big long polypeptide, one wrong amino acid, what's the problem? Well, that can make all the difference. In some proteins, it doesn't have much of an effect, but in many proteins, it can have sufficient effects that the protein's no longer the same shape as it was, and it doesn't work. And this specific mutation is one that happens in the production of human hemoglobin. This is a mutation that exists in humans. Where you get that mistake, you get the amino acid valine replacing the amino acid glutamic acid. in the structure of hemoglobin, and the consequence is that the hemoglobin that's made is not the same shape as the correct hemoglobin, and because it's not the correct shape, it can't bind as effectively with oxygen. And you remember from AS2 that each hemoglobin molecule binds with 4 oxygen molecules. Well, in this case, the hemoglobin is the wrong shape because of this mistake in its code, and as a result, the hemoglobin doesn't bind properly to oxygen. And this people with this mutation suffer from a disease, sickle cell anemia, where their blood doesn't carry oxygen effectively. And just made a wee note at the bottom, I said there, if you get a substitution mutation in the third base in a sequence, it won't have any effect, or it may not have any effect if the code is degenerate, because the third base is not important. It's the first 2 bases that are important. And for most amino acids have several triplets that code for them, and the third base is the unimportant one. The other kind of mutation is a deletion, a base deletion. Base deletion simply means delete, you leave out a base, and you don't replace it with another one. So base is completely left out in the sequence. So you'll see the original sequence in this case. Sometimes this is called a frameshift mutation. You'll see in this, the original sequence read TAG, that was the triplet, and then TGA and AAC and so on. Well, in this case, the G has been left out. The G base is missing, and you'll see down below that if you leave the G base out, what you have to do is still read the code in terms of 3 bases. So you have to go TA and take the T from the next triplet, and you can see the effect of that. It just so happens in this case you get the same amino acid isoleucine to begin with, but you'll see then that every subsequent triplet has been changed because you've moved a base from the start of each triplet to the end of the previous triplet. So the effect of a base deletion is very much greater than a substitution, because in a substitution you only just change one amino acid because you've only affected one triplet, whereas in a base deletion every amino acid, every triplet after the deletion is affected. So every amino acid after the deletion is affected. And this is a mutation that happens in humans as well, and it's one of the kinds of mutations that happen that give people a condition called cystic fibrosis. Cystic fibrosis is quite complicated. Different people have different degrees of it, and they have different numbers of deletions. But there we have a deletion mutation. A single deletion there can cause you to have a problem with the production of mucus, which is about cystic fibrosis. So just to a question relative to that, or relating to that, is question 1D. That talks about different kinds of mutations. Base deletion. It says a base deletion can have more significant consequences in terms of the final protein produced than a base substitution, which may have little or no effect. Base substitution may have little or no effect. Explain the reason for this. So you basically say a base deletion means that every subsequent triplet is also affected, and that means every amino acid after the mistake is also wrong. Whereas a base substitution, only one amino acid is affected. And if only one amino acid is affected, then that has less effect on the protein. And then it says a base substitution may have no effect, little or no effect, no effect. That's because if the mistake for a base substitution is in the last of the 3 bases of the triplet, then it may have no effect at all because the code is degenerate. The last base, you can swap the last base around and have any last base and you still have the same amino acid, so it has no effect then. So overall, what we see from that is base substitutions, both these kinds of mutations are dangerous. They can cause illnesses and diseases. But the base deletion is much more dangerous one. Base substitution may have an effect or may not have an effect. Okay, so that's that. Now the next thing we move on to is a major part of the this section, which is about how DNA codes for protein synthesis. And the first bit we come to is first of all on page 10 there we have what I describe as the big picture. And the big picture is kind of illustrating the role of DNA overall in the making of proteins. And you did a bit of this at AS. You can see on the left there you have the nucleus. In the big picture you see the nucleus, and you see the DNA inside the nucleus. And you can see there that the DNA is into the nucleus is kind of unzipping. the two strands of DNA are separating. And you have two strands. Remember the two strands in the DNA are with the template strand and the non-template strand, or the coding and non-coding strand. And you can see the coding strand, then our template strand, is being copied onto another molecule called messenger RNA. And that messenger RNA then is going out of the nucleus. And that process of copying the DNA onto the messenger RNA is called transcription. And once you have the messenger RNA produced, then the messenger RNA is a single-stranded molecule, a short single-stranded molecule. Essentially, it's a copy of a single gene. So it's like a photocopy of a gene from inside the nucleus. And the messenger RNA can leave the nucleus and go out into the cytoplasm. Where does it go to? Well, it goes to where proteins are made. Proteins are made at the RER, rough endoplasmic reticulum, and specifically they're made at ribosomes. So that piece of messenger RNA goes out and attaches to a ribosome. And then the ribosome, as it were, reads the code on the messenger RNA. It works its way along the code, and as it does that, as the ribosome moves along, you get the appropriate amino acids coming into position opposite their corresponding codes. Now, just to say that when the DNA triplet on the DNA molecule on the template strand is copied onto the messenger RNA, the 3-base sequence, the corresponding 3-base sequence on the messenger RNA is called a codon. So the 3-base sequence on the messenger RNA is called a codon. The 3-base sequence on the template strand is called the DNA triplet. Okay, so you have triplet, codon, and so on. The process then by which the ribosome reads the code, reads the DNA code and turns it into a protein, or causes a polypeptide, that's translation. So you have transcription followed by translation. Transcription happens inside the nucleus, translation happens at the RER, at the ribosomes. Okay, so we're going to look first of all at translation. And by the way, either translation or transcription gets asked every year. So it tends to be that you get transcription one year and then translation asked about the next year and year about and so on. Some years, I think the most recent, in the most recent exam, this was the essay question. And I'll just refer you to that essay question now. As we're going through transcription and translation, you'll see that question 10, the essay question, was from 2019's paper, and it's a whole essay question asking about transcription and translation. Okay, so what are the key points? Typically, if you get asked about transcription, questions 3 and question 4 there are fairly typical. And typically, you get 4 marks to describe. You get a diagram, and you get 4 marks to describe what's happening in the diagram. And basically, this is transcription. And essentially, you can see in that diagram that the 2 DNA strands have unzipped, they've separated. Hydrogen bonds have been broken, and that one of the 2 strands, called the sense strand, is then acting as a kind of a template to form the messenger RNA. So, if you're going through the steps, and you'll see that diagram in question 3 then essentially is the same as the diagram I have in the notes here. And you'll see the steps I've said. The first step is that the enzyme helicase causes the hydrogen bonds between the bases to break, and those hydrogen bonds. And we're dealing here now with a short section of the DNA, and that short section of the DNA is a gene. So basically, the helicase causes hydrogen bonds within one short section of DNA, a gene, those hydrogen bonds to break. And you can see the DNA molecule unzips as hydrogen bonds break. One of the 2 strands that you get then, one of the 2 DNA strands referred to as the template strand. And it's on the template strand that you get the messenger RNA strand formed. You could say this, you could say the template strand acts as a template for the formation of the messenger RNA. And then you can say there's complementary base pairing. Messenger RNA nucleotides line up alongside the template strand in a complementary fashion. Question 4 maybe gives a clear picture of that. You can see in question 4, you can see the messenger RNA nucleotides here coming into position. You can see there's a whole lot of messenger RNA already made there, and then there's more messenger RNA nucleotides coming into position. And you see the messenger RNA nucleotides come into position opposite the template strand, and there's complementary base pairing. And you need to see what you mean by that. you need to say that, for example, if there's an A base in the DNA template strand, then the complementary messenger RNA nucleotide has a uracil base. And equally, if there's a G base in the DNA template strand, then you get a C base. You get a messenger RNA nucleotide with a C base. But bear in mind, these are messenger RNA nucleotides that are coming into position. You can't just say nucleotides. You have to say messenger RNA nucleotides. Now, once the messenger RNA nucleotides are, as it were, lined up in position opposite their complementary DNA bases, then you have another enzyme in action, and that other enzyme is called RNA polymerase. RNA polymerase. And what it does is it joins individual nucleotides to each other. So it sticks the messenger RNA nucleotides together by what are called phosphodiester bonds. It involves condensation reactions. The RNA polymerase does not stick a messenger RNA nucleotide to a DNA nucleotide. That would be making a hydrogen bond. The RNA polymerase makes the nucleotides stick one to the other to create the messenger RNA strand. So that's a very important point, and they'll often ask that question to see if you really understood what's going on there. So RNA polymerase moves along the template strand, causing RNA nucleotides to bond to each other by condensation reactions, creating phosphodiester bonds. And that continues until you get the whole messenger RNA strand formed. Now, at this point, the messenger RNA that you've got is referred to as pre-messenger RNA. And the thing about it is it contains non-coding bits. Bear in mind that the original DNA template strand, which was effectively your gene, it contained non-coding bits. And so when you copy it onto the messenger RNA also has non-coding bits. And what you have to do now, you've got this long piece of messenger RNA, you now have to remove the non-coding bits to be just left with the coding bits. And you'll see there in that in item 7 there on transcription, you'll see that the pre-messenger RNA then has the non-coding intron regions removed. And when it does so, you'll see that it's now much shorter. So you originally have the original piece of messenger RNA with that length, but then when it had all its non-coding triplet sequences taken out, it was much shorter. Now there's quite a good question relating to that. It's question 9. It relates to that single aspect. So you've seen question 9. In question 9, first of all, it shows you two charts. On the left-hand side, it shows you the bases, the amount of each base in the original DNA. That was the template strand. And then it shows you the bases in the messenger RNA, and you kind of have to match them up. And you'll see here in the DNA there was 4 adenines. Sorry, this is in the DNA template strand there was 4 adenines. And over here you'll see that base Z has 4, and that's in the messenger RNA. So what base would you have if you have adenine here? Base Z must be uracil, all right? And so on, and you work across it and you look at the amounts and you figure out what the 4 bases is. So Z would be uracil, W would be guanine because it matches up with cytosine, X would be adenine, and Y matches up with guanine, so it's cytosine. Okay, so there you go. You can check through it. Now, it's this second part that I think is quite interesting, because it says now, after transcription, before the messenger RNA is used in translation, the messenger RNA is modified. And it shows you the amount of bases that's present in the modified messenger RNA. And you can see there's much fewer bases, because originally there was a lot of bases. There was about 2 and 5, 7, and 3, 10, 4, 14. It was 14 bases long. Now it's just 1 and 4, 5, and 2, 7, and 3, 10. So it's lost quite a few bases. And basically said, what's happening during the modification? And basically said there seems to be a lot of bases gone. And basically what's happened here is the introns have been removed. The non-coding parts of the messenger RNA molecule have been removed. And that's the answer to that up there at the bottom. 1 mark just say the non-coding regions have been removed. Now, so you've got your messenger RNA, you've got your messenger RNA made now. This is transcription. So the messenger RNA is leaving the nucleus, going out through a nucleopore, going out to a ribosome where it's going to be read. on the next page here in the notes, sorry, I don't have page numbers, you've got this table. And what this table allows you to do is to take any piece of messenger RNA and figure out what amino acids it codes for. So let's just see if we can get an example there. So yeah, if you want to, if you turn maybe go to question 1, you'll see that. So for example, in question 1, you see this here, this is the messenger RNA, this long strand. And so for example, if you said, by the way, each 3 bases in the messenger RNA is called a codon. So if you want to find out what amino acid a codon codes for, take for example the messenger RNA codon ACC. Do you see it there? ACC. And we see that it codes for an amino acid THR, that's short for threonine. We can use this table to figure that out. So if you go to this table and read ACC, what you do is the first base was A, second base was C, and the third base was C. And there you see it. There's the amino acid threonine. So you can take any base sequence, any codon on the messenger and you can figure it. And notice there's notice that threonine could be ACU, ACC, ACG. And pointing to the fact that threonine, and pointing to the fact that the code is degenerate, that the original, there was 4 different possible original DNA triplets. That's pointing to the degenerate nature of the code. So that's what that table allows you to do. You generally don't get asked that table. It's considered to be a bit too easy, but you have to sort of basically understand the idea of it. So on we go. We're moving on quickly now to translation. So and in translation, then you're going to need other molecules. You're going to need a molecule called a transfer RNA. And the transfer RNAs, in a moment or two, we'll see that the transfer RNAs is just another RNA molecule that's involved in effectively bringing the amino acids into position during the process of transcription. And you can see there, you can see there's a page devoted there to transfer RNA molecules. They're made in the nucleus. That's that page there with pictures of transfer RNA molecules. They're made in the nucleus, and each transfer RNA is a single strand of RNA. It's got an amino acid at one end and a code at the other end. So this one here's got a code UUC, and this code here at this end of the transfer RNA is called an anticodon. And basically what's going to happen is that anticodon is going to match up with the messenger RNA codon during translation, and that's going to bring that amino acid alanine into position. So there are 20 different transfer RNAs, because each different transfer RNA is attached to a different amino acid, and each transfer RNA has its unique anticodon. There's a question somewhere in the booklet there asking about the role of the ribosome. On page 13 in the notes there, you'll see there's 3 marks talking about the role of the ribosome. Consider the ribosome a bit like an enzyme. Not an enzyme, but it's an organizer. It controls the production of the protein. So basically, essentially, what it does is it brings the transfer RNA code from the nucleus together along with brings the messenger RNA codons from the nucleus together with the transfer RNA anticodons, so that the amino acids are brought into position in the correct order, and then they're joined together to create a polypeptide. So questions on translation that are quite common. So I'm looking on page 13 of the notes, and I'm going to refer you to 3 questions. Question 1, question 9b, and the essay question 10. So question 1, question 9b, and question 10. And those all contain questions about translation. Typically on any year, you tend to get a 4-marker question on either transcription or translation. So the typical translation question is 4 marks. Then you'll see question 1 is 4 marks. And then if you go to question 9b, you'll see once again it's 4 marks. So essentially they want you to come up with 4 points to describe translation. And you'll see that on the essay, essay question 10, they give you quite a few more marks. But essentially you're looking for 4 points. Now, which question am I going to look at? Well, I'm going to look at question 1. I think it's got the easiest diagram to look at. And you see in question 1 there you've got the ribosome, and you'll see that I've drawn a big a second position for the ribosome, but the starting position for the ribosome is there. That's its starting position. And you'll see that the ribosome, you can, if you draw a line down the center of the ribosome, down the very center of it, you have two sites, as it were. And the site to the left is called the T site, sorry, the P site. And the site to the right is called the A site. P is short for peptidyl and A is short for amino acid. You don't really need to know those words. In fact, you don't necessarily need to know that the two sites, the P and the A site, except I noticed in the essay question, there's some marks for knowing that. So it tends to be in these four marker questions that you can get away without mentioning the P or the A site. But I see in the essay question there's marks there for knowing the two. So where do we start here? If we were answering this question, here's where we would start. The first thing you'll notice in this question, question 1 and question 9, if you read the question very carefully, you'll see that it says, and if you look at my notes, you'll see I'm making the same point. You'll see it says, use the diagram, describe precisely what happens in the next step. And the key word there is the next step. In other words, what they're asking you is they're not asking you to describe what's happening in the picture. They're asking you, what's going to happen next? Tell us what's going to happen next. And here's what you have to say. And your starting point should be, the next thing that's going to happen in this picture is that the ribosome, which is up there, is going to move to the right. Big arrow, move to the right. And it's going to move to the right by 1 codon or 3 bases. So that's your first mark. So you start off by saying, ribosome moves to the right by 1 codon, or you can say by 3 bases. Okay? That's your first mark. And you see I've already moved the ribosome. I've done a big, like there's the ribosome in its new position. Now what you can say to get an easy mark is, do you see this transfer RNA here? It's no longer inside the ribosome. This transfer RNA then leaves because it's no longer needed. It originally brought the amino acid lysine, but your second mark you can say the transfer RNA with the anticodon UUC, or you can say the transfer RNA that brought lysine now leaves the ribosome and goes back out into the cytoplasm. And effectively what it does is it just goes and picks up another lysine and waits to be needed again, as it were. So there's the second mark. So the first mark is ribosome moves to the right by 3 bases, or by 1 codon. Second mark is that transfer RNA there that has the lysine, it leaves the ribosome and goes back into the cytoplasm. So already you've got 2 marks. Now, the next thing you can say is, you see the way UCG, sorry, the base sequence, the codon UCG is now over in this side of the ribosome. So it's now this would be the A site now. And then this other side here, I'm just going to rub it out. It's now going to be the P site because the ribosome has moved. So what you would now say is something like, so basically you would say that the codon UCG, there's there, is in the A site in the ribosome. And here's what you say. Whenever it is in the A site, that causes a transfer RNA with an anticodon that matches the codon UCG to come into position. So what you say is, you say a transfer RNA with a anticodon that matches the codon UCG comes into position. And you can actually see which transfer RNA it is. It's not this one, it's actually this one. Because you see its anticodon is AGC, and that matches up with UCG. And when that transfer RNA comes down into position, it brings the amino acid SER with it. Okay, brings that amino acid SER down into position. So that's the next amino acid, SER, it's short for serine. So that's your third mark. And then your last mark is to say this. The ribosome moves to the right again. And when it does so, then there is a peptide bond formed between the SER and the THR. In other words, there's a peptide bond formed between the serine and the previous amino acid, which is threonine. So there you are. Hopefully, if you look at my notes, you get the same sort of idea. I've got something very similar. I've just used a different ribosome and different codons and so on and so forth. But you see essentially the same thing. But you notice that the emphasis on what happens next. In other words, you have to describe the moving of the ribosome along. So in essence, and you see the same in question 9, is it 9, question 9, sorry, the question 9b, it's essentially the same thing. You see in question 9b, I've simply moved the ribosome along here as well, just so that you see what happens. So in that case, in the case of question 9b, you'd see when the ribosome moves to the right by one codon, then the transfer RNA that's attached to lysine leaves, and then you would say that the a transfer RNA with a matching anticodon to the codon UUC comes into position, and so the anticodon that matches UUC would be AAG, and AAG, if you look across there, would have been that one there, and same as that one. So that's the anticodon that comes into position. That would have brought the amino acid phenylalanine in there as well. So basically you could have figured that out. But those questions are generally okay. Most students do well in them. Most students get the 4 marks. Now you'll see also that translation comes up in the essay. Well, both transcription and translation comes up in the essay, question 10. Because it tells you, it asks you just in an odd sort of way. It says, describe and explain the roles of DNA, RNA, and ribosomes in protein synthesis. Essentially, it's asking you, tell us about transcription and translation. It's just asking in a slightly different way. It's given 12 marks there, so there's quite a few more marks. So you probably need 6 marks for the transcription and 6 marks for translation or thereabouts. Okay. Now, something else just to say about that is that, well, essentially you end up with this kind of picture. You've got the ribosome moving along here, and the ribosome just keeps moving to the right hand side, and as it does so, we just get more amino acids put into position here, and eventually the ribosome then has like a big chain of amino acids hanging behind. These are all the previous ones that were fitted, so this big long chain of amino acids of polypeptide. So when the ribosome gets to the end of the messenger RNA, then it gets off, it drops off here, and you're left with a polypeptide produced. And the ribosome then could go back to the start again and move its way along, make a second polypeptide. Sometimes you get a question, there's a question there that relates to what I have on page 15, question 7b part 2. 7b part 2. You'll see it says, 7b part 2, it says, many ribosomes often work on the same RNA molecule, messenger RNA molecule. And what that's basically saying is this, that it's not just one ribosome that might be on a messenger RNA molecule. So say this is your messenger RNA molecule, like that. Then you might have a ribosome over here. There's a ribosome there, and it's getting near the end of the molecule. And it's been making, and it would have its chain of amino acids behind it. So there's its polypeptide. But what you might have is you might have a second ribosome coming just behind it. Remember, the ribosomes are all moving over to the right. You might have a second ribosome coming, and it's making its polypeptide. Then you might have another ribosome come behind. In other words, one single messenger RNA molecule can act as can be used with several different ribosomes. So you can have a whole lot of polypeptides being made at once. And it says here, in this way, large quantities of polypeptide can be made. Suggests 2 advantages of this, and you'll see in the notes that I've said you can make a lot of polypeptide quickly. So rather than just one ribosome making its way along here, and then it has made one polypeptide chain, and then it goes back and it makes another one. If you have a whole chain of ribosomes, all one behind the other, a bit like train carriages or cars, each one making its own polypeptide, then you can get a wide lot of polypeptide made quickly. So you can get faster production of polypeptide. The second thing is that this messenger RNA strand, you don't need as many messenger RNA strands made then. In other words, you don't need to copy so much messenger RNA. One copied piece of messenger RNA can be used with several ribosomes. Whereas if you needed a messenger RNA strand for every ribosome, then that would require a lot of production of messenger RNA strands. So it means there's an efficiency of, I've said here, fewer messenger RNA molecules are needed to be transcribed in the cell. You can use them in multiple ways. Now, Earlier on there, I was talking about genes, and basically defined a gene as a length of DNA that codes for a single polypeptide. A gene is enough basis to code for one single polypeptide. At that point, I probably should have said to you, how many different genes do you find inside a human nucleus? How many, in the human DNA, how many genes are there? It's thought to be somewhere in the region of 21,000 is a figure that's often quoted. There are 21,000 different pieces of sections of DNA, each coding for different polypeptides, 21,000 genes. Now, the other interesting thing about that is that those genes are present inside every cell in your body. So every cell's got all the DNA. So a skin cell has got 21,000 genes. A liver cell, a brain cell, they all have, any single cell has all the code, has all the genes. And the question then you might ask yourself is, does every cell need all the genes? Because genes code for proteins. Does every cell need to be making 21,000 different polypeptides? And the answer very obviously is no. So whilst every cell in the body has all the code, each cell only needs to use a small number of those genes. I mean a small number, but it doesn't need to use them all. It needs to use some of the genes. So for example, a skin cell only uses the genes that are needed to make it a skin cell, whereas a liver cell needs to use a different bunch of genes to do what a liver cell does, and so on and so forth. And this idea, I'm just reading at the bottom, page 15, very important words there. It said, when a cell does use one of its genes to make a protein or an enzyme, that gene is said to be expressed. So whenever a gene is used to make a protein, in other words, whenever a gene is transcribed and translated to get a protein, that gene is said to be expressed. And the process is known as gene expression. And I've said here, because each human cell has a unique function, each human cell has its own function, each cell only makes those proteins that it needs to make. So therefore, this means that each cell only has certain genes expressed in that cell. In other words, only certain genes within any cell are transcribed and translated. The rest of the genes are not transcribed and translated, and often they're said to be switched off. The genes that are transcribed and translated are said to be switched on, as it were. The idea of switched on, switched off. In the next section then, epigenetics deals with that. Epigenetics is a study of essentially of how genes can be switched on and switched off. That's not a good definition of it. You'll see there the questions that ask about this question. I think question 10b was the first, so it's the second part of the essay question. Question 10b was the first question was asked about this. And you can see, just to notice that question 10b gave you 6 marks. That's about the size of this topic. It's worth about six marks here. So 10b, it's basically asking about epigenetics. It's also asked about in question 6. So I'm referring here to question 6 and question 10b. Question 6 and question 10b. Question 6 is a more recent one. Question 10b is about 4 years old. Question 6 is about 2 years old. In each case, you'll see in each case they ask you to define the term epigenetics, so you need a definition. It's the study of changes in gene expression that don't involve changes in the DNA-based sequence. It's basically saying it's a study of how what you get from a gene is affected by something other than a mutation. So if you had a mutation, then a gene gives you a different protein. If you have a mutation, the gene gives you a different protein. But this is looking at situations with genes where you get more or less protein or no protein at all based on something else. It's nothing to do with changes in the DNA sequence, nothing to do with mutations. And I basically set that out again. I've said each gene, only some genes are expressed in any particular cell. Now, the question is, and the general idea is that you say that genes that are expressed are said to be switched on, and genes that are not expressed are switched off. So the majority of the genes in a cell are switched off. Now, scientists are then studying how the genes can be switched on and switched off. And this falls under the heading of epigenetic modifications. the bottom of the page. I've got a page number, the Epigenetic Modifications, and there are two of those. There's something called DNA Methylation and Histone Modification. And it's usually DNA methylation that gets the most detail, and so far it's got all the marks. The histone modification is not so well understood, and so it tends not to be asked about in exam questions. If the DNA methylation has got a lot of marks. Now, I always say to students, DNA methylation, you should think about DNA methylation, you should think about it as in a negative fashion. What do I mean by that? Think about DNA methylation as a way in which genes are switched off. DNA methylation is a way of switching off genes. How do you switch off a gene? You can switch it off by a process called DNA methylation. And I've said there it stops the gene expression. In other words, it switches the genes off. And what it involves is methyl groups inside the cell. Methyl groups. And methyl groups are that there carbon with 3 hydrogens. That's a methyl group. And what we discover is that when these attach to a template strand of a DNA of a gene, that they stop that gene being transcribed. If you can stop a gene being transcribed, then the gene can't be translated, because if you stop the gene being transcribed, you don't get messenger RNA, and if you don't get messenger RNA, you can't get the gene translated. So if you look through the sequence that I have here in the notes, I've said methyl groups can attach to the template strand of a gene, therefore prevent expression. The methyl group attaches to a C base, where it's immediately followed by a G base. This is called a Cpg sequence. Now, I was looking for a question here where I could illustrate that. And if you look at question 3, you'll see there, I was just going along the template strand in question 3. There's the template strand. So it says the methyl group attaches to a C base where it's immediately followed by a G base. Well, if you work your way along the template strand there, the template strand in that question reads ATGAC. You see the 5th base, and it's a C base. And you see that it's immediately followed by a G base. So apparently your methyl group, which is a chemical grouping or a chemical, simply attaches to the template strand there. It sticks onto the template strand. The methyl group does. And when it does, it prevents the messenger RNA being formed, because when it attaches to that template strand, it prevents the RNA polymerase enzyme creating the messenger RNA strand. So you'll see there it says prevents transcription of the gene as the enzyme RNA polymerase is unable to bind to the gene, so messenger RNA can't be made. So basically the methyl group gets in the way. It interferes with the action of the enzyme that's making the messenger RNA, and because it does that, then it prevents the formation of the messenger RNA strand. Other things you need to know about this: DNA methylation is permanent and stable. Now, and you might be asking this question. If I say, for example, there's a skin cell, and that skin cell has a lot of its genes switched off, and you might say, how are they switched off? And I'd say, oh, they've all got methyl groups attached to them. The genes have methyl groups attached. You say, when did that happen, and how did that happen? And the answer is it happens as inside the embryo. So whilst you're forming as an embryo inside your mum, genes are being switched on and switched off, or it's being decided which genes are going to be switched on in a particular cell. So if a cell is going to become a cell in the embryo doesn't have any shape or form effectively, and the cells haven't. But it's in the embryo that it's being decided what a single cell, what a particular cell is going to be, whether it's going to be a skin cell or a liver cell or whatever. At that point, there's a mechanism by which, if a cell is going to become a skin cell, then a lot of the genes will be switched off that are nothing to do with skin cells. So the genes will be switched off, and they're switched off by having methyl groups attached to them. Really difficult topic, that's a difficult idea, but attaching a methyl group then switches a gene off. So I've said, to give an example, or I've said, for example, in the embryo, in liver cells in the embryo, the genes coding for liver enzymes are not methylated, because the liver cells are going to have to produce liver enzymes. So those genes are not methylated. So the liver cell can therefore produce those enzymes. Whereas in other types of cells, like a skin cell, the skin cell has the genes that would make the liver enzymes, but those genes are switched off because the skin cell doesn't need to be making enzymes like a liver cell does. So genes that are not needed instead are cell switched off. And this DNA methylation passes from cell to cell in mitosis. In other words, if a gene switched off in a parent cell, and that parent cell divides to produce two daughter cells in mitosis, then the daughter cells will also have the gene switched off. And DNA methylation then can pass from parent to child. So if a gene switched off in a parent cell, it can be switched off in the cell of the child. Histone modification is the other example, and I'm not actually going to go into it in detail at all, because if you look at the essay question on this, that was question 10 part B, you'll see that there was no marks given for histone modification, other than knowing that it existed. All the marks were for DNA methylation, and you can read through the marks there on that question 10B, and you'll see the sort of marks you got, and see the sort of things you're getting marks for. You can see there I've said both methylation and histone modification regulate gene expression. In other words, they're ways of switching on and switching off genes. And then you would ask the question, well, what's the advantage of having genes switched off in a skin cell that's not needed by the skin cell? And you would say basically the advantage is that genes are only switched on, or they're only switched on in those cells where you need to produce that particular protein. And that means that there's economy and there's efficiency. You don't have cells making proteins they don't need to be making. So because if a cell's making a protein that it doesn't need, then it's using up amino acids to make that protein. And those amino acids could be used for some other protein. So you'll see the answer to that at the bottom of page, the bottom of the second last page there, talking about the efficiency of the efficiency of this process. The other question I think I've pointed you to is a more recent question, and it's question 6 there, which you can look at the answer scheme in it. It's a recent question. Let me just look there. So first of all, it asks you about the definition of the term epigenetics, and then it shows you what it shows you here up the left hand side is gene activity. There's another way of saying gene expression, and along the bottom you can see the percentage of CG sequences with methyl groups attached. So basically that graph tells you, is basically telling you that the more CG sequences in the template strand that have methyl groups attached, the lower the gene activity. In other words, the less gene expression you get. So the two are inversely proportional. And then it goes on just to ask you how does methylation affect gene expression. So it's basically asking you to look at that graph and see what happens. So basically you're seeing as the percentage of CG sequences with methyl groups attached increases, gene expression decreases. And you'll notice there that you'll see that if there's none of the CG sequences, so over at the origin there, you see if there's no CG sequences, then the gene has 100% activity. And equally, if you get a very large number of CG sequences attached, that's over at this end of the graph, you see if there's zero activity in the enzyme. In other words, zero activity in the gene, in other words, the gene switched off. And then you get more marks than the rest of that for just saying how does the Cpg sequence switch off the gene, and you'd say that, or sorry, how does the methyl group, and you'd say the methyl group attaches to C bases in the template strand, where they're immediately followed by G bases, i.e. Cpg sequences, and that gets in the way of the RNA polymerase enzyme, and that prevents the formation of messenger RNA. And then down the bottom there is where they answered. And then just finally on the back page, I'm just finishing here now. The back page is just, there's no questions been asked about this, but this is a modern science. This is something that's really only been discovered recently, that genes are switched on and switched off. And it relates to, there's a lot of research going on with regards to cancer. Just this relates to a particular group of genes called oncogenes. You'll see in the middle of the back page there, you see oncogenes are special genes inside cells that allow the cells to divide. That when those genes are switched on, the cell divides. Now, most cells in your body shouldn't be doing cell division. For example, a skin cell shouldn't be doing cell division. A skin cell usually falls off and dies. It shouldn't be doing cell division. and it doesn't do cell division because its oncogenes are switched off. But under certain circumstances, maybe to do with radiation or something you're eating or cigarette smoking or whatever, the chemicals or the radiation causes those genes to be switched on by accident. So basically the skin cell then has genes switched on that allows the skin cell to start to divide. And when skin cells start to divide where they shouldn't divide, you get a lump of skin cells, you get a tumour, and that tumour becomes a cancer. So you can understand then how scientists are interested in this, they're interested in how you can control, how you could stop cell division, how you could switch off these oncogenes in skin cells doing cancer. So anyway, that's a quick rush through that. That's the start of the DNA topic. It's a very big topic in Module 5, and it continues on, and I'll look at the rest of it the next time, the rest of the DNA topic the next time. Okay, thank you very much. Hope that was helpful. Bye.